Yeah, I'm going to have to go back and forth, I'm afraid, between Google and other things, but this won't be too bad. So
Sandy, did you suggest that people add questions to the chat?
Sure, sure. You can feel free to add questions to the chat and we'll get to those after the talk.
Thank you.
Okay, so I just want to remind everybody that we have the schedule for the project talks is online at our website. You can find it if you go to the front page and you go to events, and it's right here. So it takes you to all of them. And then we're putting each event as an individual item later so we can expand on resources and information about the projects and about the talks. So this is our talks for today. And I'm going to talk about developing cures for antibody engineering. Before I launch into this a little bit, with each of these, we're putting links to the ATE project so you can learn more about the ATE project and see what people are doing. And we also have links here to our hackathon site. And we have a really fantastic blog by one of the student participants describing his experience in the hackathon, which I'll talk a little bit about later. And now, a video. Kind of get you in the mood. And we have Todd Smith to thank for the really catchy video. So I'm going to stop sharing that now and go on and share my presentation. Okay. So play, there we go. All right, so the title of my talk is Developing Cures for Antibody Engineering. We have a lot of people on our team and a lot of them are present here today. There's Todd Smith, who made the video. There's Maggie Bryans, who has made all kinds of contributions to the community and collaborates with Wistar. Feather Ives, Erin Kamajaya, Sheila Vemu, who is here today also, and Alyssa Dillman from the NIH Data Science Program was part of it as well. Okay, so what I'm going to talk about, just a little bit about the goals of the grant, talk about what we did in the hackathon, our results, the projects, and the next steps. So our goals in the grant, we have two major goals. One is to develop modules that support undergraduate research projects in antibody engineering. The other is to look at the use of hackathons as a strategy for doing this. So why antibody engineering and what is it? We chose antibody engineering, one, because there are over 100 antibody drugs now that have been okayed for use by the FDA. Antibodies are one of the most important tools in research and medicine. We use them for, as you saw in the video, we use them for diagnostics, for research tools, for medical treatments. So they're everywhere. And students that know something about working with antibodies can use that skill, leverage that skill and that knowledge to get jobs, because there are companies that work with antibodies all over the world. we also like the idea of antibodies because not only can antibody engineering kinds of projects lend themselves to working in the lab, but they also can involve computational work. So we can do sequence analysis, we can do homology modeling, antibody profiling, we can characterize binding sites and predict epitopes. And there are a lot of different kinds of techniques that can be involved with antibodies, from making hybridomas and cloning the genes and measuring all the antibody properties to isolating RNA, doing PCR, vectors, CRISPR, producing antibodies. There's a lot of places we can go with all this. So why a hackathon? Again, hackathons, unlike traditional professional development workshops where you're learning a skill, hackathons involve creating something. And we really like that idea because if we're making tools and projects that people can use in undergraduate research, there has to be a creative element. And that is pretty exciting. So hackathons, in a way, Alyssa Dillman describes this, that you think about them as a kind of pyramid or a triangle. One corner, one aspect is the creativity part, building something, making something. One aspect is that you're learning how to do things and learning about new things. And the other aspect that is also really important is that you're also building a community and networking and learning how to work with other people. So what did we do in the 2022 hackathon? We had, I guess at the beginning, let's see, we had 70 people applied from all over the world. We had to narrow that down because to make it a little bit more manageable, but we ended up having about 48 participants. On the last day when we were doing presentations, being a Sunday morning, we ended up with 36 people there. We developed some different project ideas, and I'm going to talk about results and the next steps. So first of all, a little bit about the hackathon and how this worked. It was a 4-day event. We started off with kickoff talks, like what do we expect to do? What are the logistics? What are the teams? Talked about antibodies and IC and 3D. Dr. Erin Dolan, who is like the expert on undergraduate research, came and talked about cures and what course-based undergraduate research experiences are. Marcus Mendez from La Jolla Institute talked about the immune epitope database. David Bloom talked about antibody manufacturing, and Francis Weiss Garcia talked about validation of antibodies and why that's a really important thing to do and why that can be different in different contexts. So after the kind of the preparation talk, so that everybody knew a little bit about the subject, all the teams met with each other and started working on planning out their projects. And the rest of the day was involved in working on the projects. The next morning, all the teams met and gave presentations about what they were going to work on for the next couple days. The writers met, and we had open work time. The next day, Saturday, was kind of the same thing, except we just had kind of updates about what was going on. The other things that we did in these is we learned how to work a little bit with cubes, which we're going to be learning about a lot more. We also had demonstrations of different kinds of software that we'd be using in the project or that might be used by different projects. For example, Jiao Wang from NCBI demonstrated new features in ICN 3D. I demonstrated how to use nextstrain.org, a really important program, web program for looking at phylogeny of viruses. And then the last day we had presentations. So what were our results? I just love this. This is from the blog that Nelson wrote. He said, "This hackathon taught me that it is possible to create a community out of complete strangers and have meaningful conversations about our reality and how science influences that conversation." So great. He's a business student who participated in the hackathon. He's from LA Pierce Community College, and he was fantastic. He gave their presentation for them, which was really wonderful. So how would people rate it? You can see that this is the last day. So from 36, we had 32 people responding here, and ratings were pretty high. I didn't have time to go through and get all my favorite quotes out of here, but can see worked out pretty well. And almost everybody said they learned something new from the experience, which is wonderful as well. And they also worked outside their comfort zone. In fact, speaking of comfort zones, one of my favorite quotes, we had a question about what would you advise future hackathon participants? And one person advised that they should probably keep a cup next to them during the initial talks because they wouldn't want to have to leave and use the restroom because they wouldn't want to have to miss anything. I think we might plan in more of a bio break on the first day after reading through that comment. So what projects did we work on? The 5 projects that we focused on for the hackathon were projects that were suggested by our advisory board. One was working on IC and 3D for education, and this was led by Todd Smith, and Jayao also helped out with this. Another was looking at whether or not anti-SARS-CoV-2 antibodies would work against different variants. Sheila Vemu led a project working with the immune epitope database, and we had people from the immune epitope database also helping out and participating. Maggie Bryans and Feather Ives led a team to break antibodies, which is really neat. And Aaron Kamajaya is working on developing techniques for doing affordable antibody engineering, which was also pretty fun. I just want to show you one thing quick that was kind of related to our project. And what this involved is, so we're looking at a lot of different antibodies and how they might function and how they might protect us from SARS-CoV-2. So we also need to think about where SARS-CoV-2 contacts the ACE2 receptor. So this is just an example of one of the things we can do with aligning structures and looking at how this works. So you see here we have the receptor binding domain from the SARS-CoV-2 spike protein. Here is ACE2. And here is one of the antibodies, one of the commercial antibodies that people work with. And you can see how this might block binding because it's actually binding in the very same spot. So we can understand why antibodies can be neutralizing. It's not true for all the antibodies that protect against SARS-CoV-2, but it is for this one. Okay, so now I would like Sheila to just say something quick about the IEDB project. And do you have your slides, Sheila, or should I pull them up? Sheila, do you have your slides or should I pull them up?
I have my slides.
Okay, cool. Then take it away.
All right. Hi, everyone. This was a great experience. I hope I'm able to share screen. Yes, I can. So I sort of wanted to start with this idea in mind. Is one thing that the hackathon really contributed was our team to work together, and it also allowed us to really keep it structured, and it was super short and immersive in nature. And the best part of our team, we had experts from IDB, but we also had people from different backgrounds. We had 2 students, we had high school faculty, community college faculty, as well as Christy was with us. And it was just a great experience because there's different expertise that you can actually build into a hackathon. And what we also figured out is post-hackathon, we could sort of think about skill development. We could think about ways to utilize and build capacity. And the knowledge transfer that seems so difficult in a bench situation in a lab seemed quite seamless in a hackathon format. So this is what we worked on. We did what we call as Flirs. This is the key acronym we gave for cures for the flu. Our team,
Sheila, we're not seeing we're seeing your screen, but not your presentation. Not your slide. Okay.
Can you see it now?
Okay.
Oh,
Yes.
Yes. Okay. So Flirs Cures for the Flu. This was the acronym that we had for our team. Our team is made up of Valerie, Christine, Miko, student, Yasmin, another student, and Christy. Thank you for being part of our team. And of course, I had some expertise help from IDB. So what we worked on typically was a piece that was really interesting. And that was to look at what can infectious diseases do and how can we sort of fit them into an IDB protocol. So we built up an abstract. So our first day was all brainstorming and we came up with an idea of flu. We say, what can we do with the flu? And we built up some kind of a project. And this is the project that we are looking at. Because many of our students in our community colleges, because of remote teaching, we're not able to do a lot of research in the last couple of years. So we thought about what is one way we can actually put together a research project. And this is one of this. And I wanted to go over, this is a project that we created as some lesson plan that we could apply to our summer programs. An example curriculum. So students were asked to create a better vaccine. Students are provided with a background on influenza and vaccines. And we decided to use the immune epitope database analyses resource. This is freely available. It's funded by the Allergy and the Infectious Disease. It's got an experimental data set of almost 1 million epitopes of antibody and T cell epitopes that are actually cataloged from multiple animal species. So their research interface is pretty easy to use. We had some learning outcomes set up for our students. What would they know? We started to grade it from basic terminology all the way up to using the epitope analysis tools, and for students to sort of distinguish between linear and discontinuous epitopes. Because when we are teaching some of these courses, it's really difficult for the students to understand granularly what's really happening when you're looking at different epitopes. We also worked on some of the research database, and I'll quickly go through it and I'm going to wrap up so that Sandy can go along. Is that we also have an immune profiling piece that we created with the immune epitope database that actually worked to our advantage. So with that, back to you, Sandy.
Thank you, Sheila. And before we start our discussion, I'd like to ask that everybody please complete the evaluation sometime today, hopefully soon, because that really helps us in the planning and helps us understand, how we should structure things, and it's really informative. Okay, so questions. Let's see, I guess I can go through and look at questions in the chat. Linnea had a question for Christy about whether your industry advisory board helped in determining the assessment tool, and if you incorporate industry-related laboratory documentation.
Thanks, Sandy. I answered this for Linnea in the chat, but I'm happy to talk about it some more. We finally were able to get a workforce advisory council off the ground and meeting regularly on a quarterly basis. So they didn't help us develop the assessment tool, but the industry partners used the assessment tool, and then the members of the Workforce Advisory Council did review, the data from the pilot, and didn't have any suggestions for changing them. They seemed to think that they were, the skills, and they liked that, they were common skills across academic and industry labs. And then, yes, the documentation was definitely something I wanted to work on. So as I said, my background is in academia. So I reached out one of those industry partners, and Sandy, it was one of the ones we actually talked about because they do a lot of antibody work, integral molecular. So it was that industry partner that really provided us with lots of sample documentation. And so, we just started simple by redoing our SOPs for pieces of equipment in the training lab. So, you know, we're working our way to incorporating some more of that documentation, because I know it and that's something that we hear right from, you know, associate degree up to postdocs. When they make that transition from academia to industry, they can't believe that change in documentation amounts.
Excellent.
Thanks, Christy. Other questions if people want to speak up?
I think you answered my other question, Sandy, about projects were determined ahead of time.
Well, sort of. I mean, mostly the titles. Exactly what we would do, not so much. And some projects more so than others.
And Linnea, I may answer that because some of our tools were determined, like I knew I was going to work with IDB, and I was just sharing with Sandy that I was thinking I'm going to do something with SARS-CoV-2, but our group wanted to do something with influenza. So we sort of used the brainstorming procedure to see what is the consensus of what the group can want to do.
Maggie, what would you say? I should have described your project more too, because it's really neat.
Oh, no problem, Sandy. So our group started from ground up. We had a title and that was it. And we knew we wanted to develop wet lab undergraduate research experiences around genetically modifying an antibody so that would change certain amino acids and then change properties of the antibody. But we had to make it actually doable in a community college lab. And that was a challenge, because we can propose, you know, anything we like at any level, but it actually has to be able to be done in an undergraduate lab. So we spent a lot of time researching that, you know, off the bat, and we ended up having two different projects, one's around a GFP nanobody and another one around a therapeutic monoclonal antibody. So we actually developed two potential cures, and also using IC and 3D to determine which amino acids we were going to change as far as how it interacted with the epitope. So I learned a lot. It was great working under pressure. You know, it's always even better in retrospect. At the time, it's a little intense, but afterwards, in retrospect, it was a great experience. I think the whole team agreed. I recommend it.
Yeah, that was the most common question we got is, you're doing this next year, right? And yes, we are. Our only challenge might be just, we have to have enough team leaders so that we can scale, because we don't want to have the team so big that they can't work together and get work done in a good way. I didn't mention too, Aaron's team, they were actually in the lab working. Aaron sent me a list of stuff that he needed, and his students were working on developing their yeast screening system during the hackathon so we could get data on the last day. It was pretty exciting. And Todd, what would you say about your team?
Well, my team was pretty engaged. Ours was, you know, how do we start to adapt iScan 3D for education use? And I wouldn't say we got that far, but we did get some interesting examples of how to use it for specific things. A couple of Sheila's colleagues who teach chemistry were part of our team, and they really liked the idea of getting into a protein structure and looking at amino acids and looking at the chemistry and how you can, you know, start to get, you know, make it more relevant for students. You know, some of these topics can be a little bit abstract to the uninitiated. So people were pretty enthusiastic about that. Yeah, one of the things, you know, in addition, you know, we had Nelson, the business student, he did quite a few presentations, which was great, because he's learning words on the fly. And I was thinking, you know, with Aaron's experience, what we need to do is partner with some of the media teams at colleges so that we can live stream some of the laboratory work if we have that going on. I think that'd be kind of fun.